Hi, my name is Saif, and today we're going to be talking about the variability and homeostatic processes of neurons and networks. Now, neurons fall into a very particular type of cells that are known as terminally differentiated cells. That means that they actually don't duplicate, they don't replicate themselves. Now, another example of a terminally differentiated cell inside of the body that's extremely common is this guy right here, the blood cell. Now, the blood cell has a turnover rate of 120 days, which after that point, it is recycled, and a new blood cell is prepared from the bone marrow, and it becomes a cycle. But neurons, although they're terminally differentiated, they don't die, and they're not replaced. So when you can't replace the entirety of a neuron, you can replace the constituent parts. Now, this is the famous neuron that we all know and love. It has the axon and it has the famous soma. Now, the soma is very much responsible for that electrical potential that we love and we hear about all the time that comes from the axon hillock. But what it really stems from is the movement of ions. And now, if you want the movement of ions to go through a membrane, you must have channels. Now, depending on how many channels you have and what type of channels you have, you'll have different types of action potentials. Here is the action potential that we've probably all seen at one point in our education. But underneath is something that you may have not have seen. These are the conductances of the respective ions that really contribute to that action potential. Conductance is basically all about flow. If I say at this point over here, there's a high conductance of sodium, that means a lot of sodium was passing into the cell. And if I say that the conductance of potassium is very high in this point, it means a lot of potassium was passing through the cell at that point. Now, this slide shouldn't scare you, but if it does, don't worry. We are focused here on it just because of modeling. Now, we all heard about the Hodgkin-Huxley model of ion transfer, which was basically describing the giant squid axon. Much of what we're going to be talking today is based off the somatogastric ganglia of the lobster. Now, in this example, this is just the giant axon model, the Hodgkin-Huxley model. What we're just talking about is that how can we predict or know how much conductance and how much current is being produced? So we usually have many factors. This G would be representing the conductance if all the channels are open. The I just stands for the amount of current that is there. And here we can see the M and the H. They're the probabilities that a channel is even open. Altogether, we're trying to know about the voltage that is inside of a cell at a current time. So we got a little bit of background on some of the general processes of how neurons work and the conductances of ions, which basically create that action potential that we see. So we're looking here at two different papers. We can see that Eve Marder here is one of the authors in both of them, and you'll hear her name a lot. You can search her up online. She has a lot of great stuff looking and diving into neuroscience. So let's look at our first study. What we're going to be doing is we're going to be looking through studies piece by piece to learn small little facts and axioms of knowing the homeostatic functions of neurons. So in this first study, we're looking in the Purkinje neurons. Purkinje neurons, if you've ever heard of them, they're in the heart. They're autonomic in the sense that they have their own pacemaker rhythm, and they don't need an external neuron to fire on them first for them to shoot. So here we have cell number 1 and cell number 2. This is from the Purkinje fibers. This is their spike potentials. Now cell number 1 and cell number 2 look awfully similar. And this is just the voltage. But what if we go down and look at the actual current of sodium, the current that is being created by the conductance of sodium? We can see in cell number 1, there's far more current of sodium at negative 156 picoamps compared to the negative 41 picoamps of cell number 2. And the inverse here is there true. For the current of calcium inside of cell number 1 is negative 221 picoamps, and in cell number 2 it is negative 823 picoamps. But we see that the action potentials and the spike potentials look very similar. Now, if you look to your right, we have a similar diagram. We can see number 1, 2, and 3 have very, very similar waveforms already. But you see that the conductances, remember G just means conductance, the conductances of the respective sodium and potassium is very different. In number 2, they almost look like they're equal to each other. Number 1 and number 3, there's huge differences. But all of the spike potentials look very similar. We'll talk more about the knowledges we'll gain from these experiments in the next slide. But I want you to take a look carefully at D. In D, it is basically a summation of numbers 1, 2, and 3, and it takes the average of them. However, when we averaged out those respective conductances in those particular cells, we actually didn't get the same waveform. We had 3 simultaneous bursts in a spike potential. So we just looked at the experiment, so now let's look at what we can learn from it. Number 1, there is more than one way to reach a specific firing pattern and spike formation, like we just saw. At different variable conductances particular to each individual neuron, they can all still have the same firing pattern output. But we need to also understand that you cannot take the average of the conductances. This may tell us, and it is an indicator, that the channels may not be the focus here, because if we took an average of all the channels, activities, and conductances, we should be able to, in theory, if they were the key player, we would have the same firing pattern. But as we saw in the previous slide, that wasn't true. Now, let's remember with these new 2 lessons that we learned, let's move on to the next experiment. So with the 2 new things that we learned, let's move on to this procedure. In this procedure, we see that inside of a neuron, a shell RNA was injected. Now, shell RNA basically codes for the channels that cause the transient potassium current. So these are potassium channels. So when you're injecting RNA, that RNA is then going to be transcribed and basically converted into the protein, which is known as the channel for the potassium. Then it was left for 72 hours, and then the spike forms were compared to base. So a summary of what just happened. We saw that there was an increase of the transient potassium current. Now that's expected, because if you take the RNA is going to be converted into the actual protein. The protein in this situation was the actual channel for the potassium. However, interestingly, there wasn't any change in firing pattern, even though we changed this major conductance. And this might be because there was also an increase in the hyperpolarized inward current. And we know that the IA and the IH, the IA being the transient potassium current and the IH being the hyperpolarized inward current, basically coupled and canceled each other out. Now, if we look at this data right here, you might be able to see at A that the dark line is the shell, and we see with the shell RNA, which had the transient potassium current come out, it had that giant peak there. But if you look down to CI, you can see that the actual waveform was still very similar to the baseline. And now we'll look at the next slide to see that hyperpolarized current, which basically canceled out the shell. So again, you can see the light line. So we're looking at B this time on your right-hand side, B. So that light line is the baseline, and the dark line is the shell. And we can see that that downward peak is actually that hyperpolarized inward current. And so this is what is canceling out that activity from the IA. So here are some takeaways from that experiment that we just looked at. Number 1, neurons are always tuning themselves. We saw that the transient potassium current was increasing, but however, it was met with a hyperpolarized current. So that canceled out any of the activity, and that's where we saw the same waveform as baseline. So that also makes us and leads us to think that there must be some way that the cells are sensing, or that the neuron is sensing the changes. So this leads us to our next experiment. In this experiment, which Now, this experiment is very, very interesting because it actually uses a computer-based model to model that somatogastric ganglia. So we talked about how the squid giant axon was used for some of those very early currents and that sodium and the potassium pumps. But however, our focus here is this entire system, this entire ganglia. So we'll talk about it later on inside of the actual presentation, but I want you to look at the fact there are 7 active currents that are programmed into this software to help us basically deduce information and to run simulations. So there's a lot of math that was involved here, but what was actually realized at first, what was proposed in the beginning, was that there must be a sensor. So through a lot of math and through experimentation, through the actual simulation, we learned 2 main things. Number one, as activity increased inside of the cell, the calcium concentration rose. Number 2, the possible mechanism of detection would be a bulk intracellular calcium sensor. So anytime you have more activity in the cell, more firing, you'll have more calcium. And we'll see that in the next slide. So we can see here that in the silent stage, if we look at A here, at the silent stage where we have very little, again, GCA, which is the conductance of calcium, at very low conductance of calcium, we have very low firing. And when we get to burst and spike, we have more and more calcium conductance and you have more firing. Now the calcium is not the one causing the firing. It's inside of the cell, when you have more activity, a lot of the cellular cascades use calcium, and calcium is a measure of how much activity is actually inside of that cell. So again, we have some more lessons we can take back. Number 1, intracellular calcium is an indicator of activity. The cell can use this as a monitor. So we know that whatever is going on in terms of homeostatic processes, what it could be is that it is using the calcium as a sensor. So more calcium is inside of the cell, then through the sensor, you will have more of a transcription of, for example, different channels to bring you back to our second point here, a set point. It seems that a neuron is always trying to get back to some sort of set point. And this makes us wonder that where did this set point come from? And we'll talk about it in just a bit. So this is that diagram we just saw before in a different way. Now we know that when there is a slow and a little amount of activity, so here we see on the left only 2 spike potentials, we have very little calcium. And we have a lot of hyperpolarizing channels. And as the activity increase, what we see on the right here, we have a lot more calcium. And again, the calcium would then go to the calcium sensor, and the calcium bulk sensor would be able to then upregulate or deregulate the amount of channels that are present on the actual membrane. Now, it's also very interesting and important to remember that sometimes, even if you are transcribing and transposing more channels onto the membrane, sometimes those channels could be creating hyperpolarizing currents and not depolarizing currents. So it's all depending on what type of current comes from those channels. So just because you have more channels doesn't mean you're going to be more active. So what we basically can learn from here is that channels, like we were just mentioning, are not the focus. During development of the neuron, it was designed with a set activity point, and that is what is being regulated, and that's what we're trying to reach here. So the picture that is on the bottom here is actually a picture of a computer's neural network, obviously based off actual neurons. Now, we can see here that on the right hand side, it always bubbles down to one point. The neuron has to always try to get to one type of function, that similar spike pattern that it was programmed with. Now, in the middle, we can say that these functions, that the conditions of the cell, for example, we can say in the green box, the green circles, it could be the different concentrations of potassium and sodium conductances. And in the middle, the hidden layer, the red part, we can say these are the work and the acts of the bulk intracellular calcium sensor, where it is basically transposing more channels to either increase the amount of hyperpolarizing current or decreasing the amount of hyperpolarizing currents. So we'll get to another experiment here. Now this experiment is very interesting. What it did is that it went to the neuron and it applied tetrodotoxin, TTX. Now, if you don't know, tetrodotoxin is something that actually blocks the inward sodium current. It's actually a potent poison found in the pufferfish. Now, the tetrodotoxin would basically stop the action potential from occurring inside of the cell. And then what the experiment did is that they monitored and looked for activity, and then they re-stimulated that same neuron and they washed away the tetrodotoxin, and they wanted to see at what state that neuron was. So here, what we actually see is very interesting. Now, when the tetrodotoxin was actually applied, slowly the cell and the neuron began to increase those sodium currents, and the inward sodium currents, and it started to decrease those outward sodium currents, trying to reach the homeostasis even when the tetrodotoxin was there. Now, when the tetrodotoxin was taken off, now this neuron has already homeostatically, to some degree, compensated itself. So what happened is that when it was re-stimulated, it created several more action potential. So the burst is having many more action potentials than was in the control. And this is because our takeaway is that activity depression causes increase in activity through compensation, because during that time you had the inability to basically have that inward sodium current. Slowly the neuron by itself compensated, like using the other mechanisms we mentioned before, to basically come back to that fine-tuned spot which it was programmed with during its development. So we'll go here into one more very interesting, this is our last study. So we're going to talk a little bit more again on the actual program that is simulating the actual neurons. Now, this is the somatogastric ganglia of lobsters. So usually in the human brain, we're talking about billions of synapses. So it's very difficult to basically compute all of those things, and sometimes nearly impossible. But luckily for us, for this lobster, there's only about 36 neurons to compute. So the ganglia is obviously the collection of the cell bodies, and so we have some main focuses here. So if you turn your attention to the right, you'll see where the different waveforms in red, blue, and green. AB is going to be standing for the anterior booster. The PD is the pyloric dilator, LP is the lateral pyloric, and the PY is just pyloric. So these are neurons coming from different areas near the stomach of that same lobster. And this model system is used and was used even in our calcium sensor experiment that we mentioned. So using this data, what was done is that 20 million simulations were ran, and those simulations that resembled the biological parameters, meaning what was actually found in normal somatogastric ganglia of lobsters, was used for this experiment. So just a couple of things here before you move on. If you look very carefully how LP, PY, PD, AB, all of these different neurons are basically linking to each other to basically create a circuit. This is called a central pattern generator. This oscillation that you see that is basically that is in sync, that we can see when PD is on, LP is not going to be on. And similarly, PY is not going to be on. And they basically are following this cyclic pattern. And this is actually basically happening because of how they are coupled together. That's just an interesting point I wanted to mention here. So now when we go back here, so a lot of the other processes we were seeing that between two neurons that have totally different conductances, we can see they have a very similar action potential spike formation. Now, we're basically taking those principles. These are actually referring to full-on networks. So this is a mixture of the ABLPPY neuron networks together, and we're trying to see if those same principles still apply. And if you look very carefully, of course, they still apply. We see a so in the pink and the blue, we see very similar spike formations. However, if you look down to B, they are very different conductances. And this goes to show that, again, those principles that we learned before still apply to networks as a whole. So let's look at those takeaways. Number one, the principles that we learned about that there are many ways to reach a set activity level. That, for example, you have that one point that you want to reach, that one node in that neural network. However, you have many ways to get to there. You have many nodes and pathways that you can take. And we learned here that there are many different types of conductances and different amount of channels that you can have inside of that cell and still reach that same activity level. We know that calcium is an indicator of the activity inside of that cell. For instance, if you have a lot of activity, a lot of firing, you're going to have a lot of calcium. And then if you have a decreased amount of firing, you're going to have a decreased amount of calcium. That calcium binds to that intracellular bulk calcium sensor, and that can basically go on to transcribe more channels and increase or decrease activity to, again, get back to that set activity level. We learned about during inhibition, the cell and that neuron basically compensates. For example, in the tetrodotoxin, which basically blocks the inward sodium current, as a compensation, that neuron basically worked very hard to try and regain some of that inward sodium current, and while at the same time it basically stopped outward currents from occurring. Now, what we learned is these are from neuron to neuron, just an individual neuron. These are intrinsic properties of the neuron. However, when we just gave the example in number 5, we're talking about a network here. The principles still apply. It's all about there are multiple ways to reach one activity level. There are multiple ways to basically potentiate the synapses between neurons, because that's the connection between them. Between two neurons, we have synapses. So calcium there also becomes an indicator of activity. And there is compensation for inhibition. So during long-term depression and long-term potentiation, these are the ways that the synaptic structures of the networks are basically allowed to reach homeostasis. So that concludes this presentation. I hope you learned a lot. So ask the questions, and is there anything more if you want to learn, go ahead and you can just ask that away. Anyway, have a great day.